Hello, my name is Amanda Thomas, and I'd like to welcome you to A Scientist Walks Into a Bar. This podcast features recordings of talks given at Science on Tap, a series of science lectures held in Portland, Oregon, and Vancouver, Washington. It's been an embarrassingly long time since we've produced an episode, but we have two pieces of good news. The first one is that we've continued to hold lots of events since the last time we posted an episode, and we have recordings of many of those talks, so we have plenty of episodes in the queue. That also means that you might be hearing talks that were given a few years ago, but I promise that the information is still relevant. The second good thing is that this time we're working with an actual audio technician who will be able to help keep production rolling along, and the plan is to release a new episode every 2 weeks. We figured that it would be appropriate to start back up with an episode about the science of beer. This is a recording of the talk Evolution Under the Influence, Alcohol and the Coevolution of Humans and Yeast by Dr. Kevin McCabe. Kevin is a geneticist who has worked in academia for years, but who now has a dream job and works in a brewery as the lab supervisor at Sail Brewing in Hood River, Oregon. Kevin gave this talk back in 2017 at the Kiggins Theater in Vancouver, Washington, and he talks about evidence that humans and yeast co-evolved throughout history, and how that affected things like civilization, our genetics, and is possibly one of the reasons that humans walk upright. Here we go.
Great, thank you very much. So tonight we're going to talk about evolution under the influence. And basically what this is about is alcohol and its impacts on the coevolution of humans and yeast. So basically this is an overview of what I'm going to talk about today. We're going to define evolution and coevolution, talk about the origins of human or ancestors' ethanol consumption, yeast ethanol production very briefly, key markers in human cultural development that were impacted by ethanol, early microbiology of yeast and how that sort of impacted us and yeast, and then talk a little bit about yeast at present. There's a really cool paper that came out last fall that I'm going to delve into a little bit. So, evolution is defined by Merriam-Webster as descent with modification from pre-existing species. Pamela Weintraub describes it as, in Darwinian evolution, the basic mechanism is genetic mutation followed by selection of the organisms most likely to survive. This is sort of the way I define it. This is my kind of favorite definition. But when we think about evolution, a lot of times we think about evolution happening slowly and progressively over time. And that's not really exactly the way it works. The key point here is this selection. You have the mutations happen first, and then you have some change in the environment that selects for organisms that are able to do better than the others, and they either survive or breed better and change the overall genetics of the population. So one of my favorite examples, I used to teach microbiology, and this is why you take your entire course of antibiotics. Doctors don't ever tell you this, they just tell you to do it. This is the why. So in any given population, you sort of have a bell curve of phenotypes. Some organisms are going to be less resistant, some organisms are going to have a little bit of resistance for whatever reason, and some are going to be highly resistant, okay? When you start taking your antibiotics and you're building up that concentration of antibiotics in your body, you start to get rid of the highly sensitive organisms and most of the intermediate organisms, but the highly resistant ones really take time and a concentration of antibiotics in your body that requires the whole course for you to get through. If you take the whole course, you get rid of all those guys and you get better and you feel better and you don't have an infection anymore. If you stop taking antibiotics prematurely, what you're left with are only the highly resistant microorganisms. And within a couple generations, they're repopulating. And given that bacteria can divide, sometimes as quickly as in 20 minutes per generation, you can run into big problems really fast. So evolution can happen very, very quickly, I guess is what I'm saying here. And it requires that selective pressure. In this case, we're talking about antibiotics. But in other cases, it can be lots of other things. So that's the basics of evolution, is selection. But coevolution is something a little bit more complicated. Coevolution happens when you have multiple organisms living closely together and interacting in some sort of way. The definition from Merriam-Webster is evolution involving successive changes in two or more ecologically interdependent species, as of a plant and its pollinators, like in the figure there, that affect their interactions. And so the funny thing is that this flower actually doesn't have any nectar for this moth. It's an orchid that is mimicking another flower that does have nectar in its base, which is why this moth has this long tongue. But these are the pollen packets that it picks up when it tries to get that nectar. So it's basically, these flowers have designed their structure in concert over time with these other organisms. And very specific pollinators pollinate very specific plants. And in this case, you can see it takes a very specific organism to get at the nectar that it thinks is at the back of this flower, at least. Plants lie. Did you ever know plants lie? It's kind of evil. Okay, so this guy. I started thinking about coevolution when I was at a talk that my father was giving. He was the president of the American Society for Human Genetics in 2009. And he gave a talk on the coevolution of humans and dogs. If that's an interesting topic, he's been following the research, and talk to Amanda afterwards or put it on your little form. Maybe we can get him to come talk. 2009 was really cool because it was Darwin's, or would have been Darwin's 200th birthday, and also the 150th anniversary of the publication of On the Origin of Species, Darwin's famous book. And again, the talk was about coevolution of humans and dogs, talking a lot about what Darwin knew and concepts that Darwin had about this. And so here we have a gray wolf, the predecessor of modern dogs. They look a lot alike, right? I generally see, my little pug coming to me with a haunch of caribou meat. But the dogs and humans have a long track record together, and we've actually co-evolved as two species. When you look at some of the archaeological evidence, you have co-location of human and wolf remains as early as 100,000 years ago, suggesting that there were wolves hanging out with humans. But when you look at the archaeological evidence of phenotypic divergence, or the appearance of what we consider to be something other than wolf or modern dog, that doesn't happen until 15,000 years ago, okay? So we're way over here. We know dogs were hanging out with humans here, but we don't really see any changes in dogs until here. So what's going on? When we take modern genetic technology and look at the sequences of DNA, we actually see genetic changes that predate this phenotypic change and co-locate in a timescale with this 100,000-year-old co-location of dogs and humans. And this sort of says that they were there, they were hanging out, and they were starting to have impacts on each other from their first interactions. And so, what impacts were the dogs having on human life? Well, the dogs probably were hanging around and using us for food. We were an easy meal, but what else do we get from dogs? We can look to modern examples of this. This is a picture from a search and rescue group. They located a child using a dog. So we use their sense of smell. We use them in hunting. We use them for protection of our homes. And I think some of the origins of this co-location of humans and dogs is that they were picking up our trash. We were a source of food for them, and an easy source of food, and the ones that were sort of pleasant enough ended up spending time with us. And we went from being their them being our garbage disposal to them being companions, intimate companions with us. In that, then both groups started to change. The change in the phenotype that we see here, that's largely thought to be a change in how humans were living. This sort of ties into around the Neolithic Revolution when humans were starting to settle down. And so it's thought that when we were hunter-gatherers, wolves were great, because that's basically what we needed them to be. But when we started settling down, we needed dogs for other stuff. So what about the coevolution? Think about some of the things that we use dogs for. We use them for their sense of smell. and their hearing. And if you look at some of the characteristics of domestication, one of the things is a shrinking in brain size. And for us, that's our olfactory sense. We don't smell very well for animals like us. And so we've basically have lost our sense of smell because we rely on dogs living with us to carry out those tasks for us. And so you have this intimate tie between these dogs and humans over tens of thousands of years that's led to changes in both. So the key point here, I guess, is that we domesticated dogs and they domesticated us. And this is going to be parallel to the story when we start talking about humans and yeast. And then another key thing here is that genetics, looking at the genetics of the organisms, shows an earlier divergence than archaeology had initially suggested. But when you look at the two together, the archaeology backs up the genetics, right? You can see those genetic changes happening here 100,000 years ago, and we see the co-location of wolf and human remains. We then can also probably look back and see some genetic changes there that are impacting phenotype. So this is our very good friend, Saccharomyces cerevisiae. They help us make beer and wine and other fermented beverages with ethanol in them. We also use them for baking bread, except there we're not using them for the ethanol, we're using them for the CO2 they produce to make the bubbles in bread and make light, fluffy loaves of bread that we like to eat. We cook off the ethanol, which seems like a waste. But the name Saccharomyces cerevisiae, basically sacca, like saccharide, sugar, myces, fungi, and what does this look like? Cerevisiae. Cerveza, right? Okay, so basically these are sugar-eating fungi that make beer. Okay, so this whole talk came about from a weird thought that I had. So I have a PhD in human genetics, and I just, a couple years ago, started working for a brewery, and I was sitting down having a beer, and the two worlds collided, and I was like, why do we have genes for breaking down ethanol? It's kind of a simple question, but it really led me to think about more interesting things. We have the genes we have based upon evolution, okay? They're not just there for fun, there's some reason why they're there. We don't make ethanol within us. So the reason we have genes for breaking down ethanol has to be because we're taking in ethanol from an outside source. And then, if you put that together, we have these genes because our ancestors were doing exactly what I'm doing now. They were drinking alcohol, okay? So, when you start looking back at the human consumption of ethanol, there's sort of two hypotheses. This is the first one, it's the lame one, I don't like it. It's the paleo diet hypothesis. And it's reviewed very nicely in this paper here by O'Keefe and Cordain. And it basically says that ethanol had no significant contribution to the Paleolithic diet, and there we're talking about 2.6 to about 10,000 years ago. That's before we settled down and started agriculture. But that ethanol consumption, we didn't really have ethanol consumption until the Neolithic, until we started agriculture and we had the food reserves to do something else with the food. We didn't need it for food anymore because we had enough of it, so we can do other stuff with it, like make beer. I don't know, it doesn't feel right to me. And so when you dig through the literature a little bit more, you come across this ethanol early hypothesis. This is basically suggesting that we know that yeast were around, and we know that yeast are present on fruit, and we know that when yeast are present on ripening fruit, they make ethanol. They chew on the sugars in the fruit, convert it into CO2 and ethanol. And so this all sort of comes together in about the same time period. You have fruit-producing plants, you have the yeast to make the ethanol, and part of the reason that these plants are producing fruit is to get animals to eat that fruit to disperse the seeds. Well, okay, so if you have ripe fruit and you have yeast on it, you can pretty much assume that there was ethanol in it. And if these organisms are eating this, our tree-dwelling, our arboreal fugivore ancestors, they must have been trying to get at that fruit when it was at its most nutritious, which is when it's ripe, which means that they're going for the same sugars that the yeast are. They want that sweet piece of fruit. The yeast are going after those simple sugars to make ethanol. So basically, these tree-living primates were eating fruit that contained ethanol. So a quick aside here about ethanolic fermentation. So we have our friends, Saccharomyces cerevisiae. Why do they make alcohol? There's this ongoing war that we never really see with microbes. Microbes are in us, around us, all over us, everywhere, all the time. You are actually outnumbered on a cell-per-cell basis by the microbes that live in and on your body, by a lot. And so all of these microbes are fighting for territory and space and food. And these yeast that want to get at that sugar in that fruit, a really good way to keep other organisms from getting at those sugars is to make alcohol. And this isn't just something that we see with alcohol and simple compounds like this. You actually see a lot of these biochemical weapons being taken advantage of by modern human society as drugs. We talked about tetracycline earlier. There it is, right there, those streptomyces. But when you look at this list of microorganisms and the antibiotics they produce, it's basically a pharmacist's cabinet. And so you have this competition between microbes, and one of the other things that we can sort of add to this is ethanol. Ethanol is a potent biocide. It's very good at killing microbes. In my lab, I use it to clean stuff because A, it kills the microbes, or most of them, and B, when I light it on fire, it kills the rest of them. So it works very, very well. So back to the ethanol early. We've talked about why and how yeast, fruit, and primate ancestors are sort of all together at this one point in time. When primates are assessing fruit, as you can see here, they're going to fruit and tree, and they're looking at it, and they're smelling it, and they're tasting it, and they're sensing the texture with these pressure-sensitive teeth that they have, and they're basically trying to decide if they're going to eat it or not. And they're picking the ones that are going to have the best nutrition, that are the sweet ones that smell good and look good, right? Something interesting when you look at this behavioral chain of deciding about eating something, you have this sort of searching behavior, contact testing, oral evaluation, and acceptance, if they're gonna eat it. And basically the visual here, they're looking at the color, size, and shape. But when they're looking at these olfactory cues, they're looking at esters, which are these very light volatile compounds. We like the way they smell. They're sort of like the fruit floral notes that fruit smells like, right? Fruit smells like fruit. It's the fruit notes, right? But then also ethanol. We can smell ethanol really well. Ethanol is also highly volatile. Like these esters, spreads in the air. What if they're using ethanol as a cue for identifying the ripening fruit? Ripening is happening, the yeast are there, they're making ethanol, so you have this as a potential marker for why the primates are going after this fruit. When they actually then come in contact with the fruit, when they get over to the tree that has the fruit in it, they are looking at for damage or color. They're looking to see if it's overripe. We'll get to that in a minute. They're then smelling to see if it has these esters and ethanol that are markers of ripeness, and whether or not the texture is right. We encounter this when we're going to the grocery store and we're trying to pick out fruit, right? We pick it up, we smell it, we feel it. They're doing all the things that we do in a grocery store. That's just their grocery stores, the trees. The oral evaluation, then, they're tasting for these esters and ethanol. This is just that sort of texture thing again. But then when you're starting to taste things, you're getting into, is it sweet? Does it have nutrients that I need? And we have sort of sense for this. The other thing that they're looking for is these antifeedants. Okay, so antifeedants are basically part of that biochemical warfare that I talked about earlier with microbes, except it's the plants trying to keep things from eating the plants. They want us to eat the fruits to disperse the seeds, but there are certain things, well, they want the right animal to eat the fruit to disperse the seeds, because certain animals are just going to drop the seed right at the base of the tree. Other animals are going to go and cache them and bury them and give that seed a better chance to germinate and grow into another tree. So they want the right organism to eat it. And so plants produce this whole arsenal of things that sort of prevent us from wanting to eat that plant or that fruit. If the fruit's accepted, they eat it. So when we look at what's going on with fruit ripening, color isn't a really good cue. Color is really deceptive. But when you look at ethanol, so here you have total soluble sugars. This is sort of a marker of nutrition. And here's the percent ethanol. When you start seeing these high levels of ripened sugars, you also see the ethanol spike up. Again, there's yeast on the fruit. They're going to start chewing on those sugars. And so ethanol is an amazing marker for the ripeness of the fruit. Stress is pretty good. It ties in a little bit with ethanol, but it's not quite as good. So basically, if you can smell ethanol really well, you can find fruit in the trees that's ripe. And that provides an advantage to you over other organisms that can't. And we sort of know that ethanol can be sensed very well. I mean, we can do tests with organisms today and see that. Primates sense ethanol better than rats, and rats basically live through their nose to find food. So why do we have these better senses for ethanol unless there's a reason for it? We have these neurological responses to tasting and smelling ethanol as well. And when we sort of put that together, I love the sort of sentence here, the collective strength of these sensory responses is consistent with the notion that ethanol plays a significant and hitherto underestimated role in the regulation of primate foraging behavior. So these primates are out there and they're climbing around in the trees. And if you think about fruit, ripe fruit in the natural environment is very transient, right? You don't just have ripe fruit out there all the time. When cherries come into season out where we live, you've got a couple weeks. And individual varieties of cherries, you've got a couple days. And so when you only have a couple days to get at that fruit before it goes bad, being able to identify it and track it down becomes very, very important. And what this is basically saying is that ethanol makes a great marker for identifying ripening fruit. Basically, being able to smell that ethanol means that you can find where fruit's ripening, go to it, and eat it, and get that nutrition. This is sort of summarizing all of that. Volatilized alcohols from fruit potentially serve an olfactory localization of transient nutritional resources. They can use the scent of ethanol to find that fruit. And the other thing is, we also know that ethanol consumed during the course of frugivory, or eating that fruit, can act as an appetite stimulant. And this isn't, okay, it kind of is getting the drunk munchies. There's this thing called the aperitif effect. And basically the aperitif effect says, or when they do studies of people, they give you a drink before they give you a meal. And then they give you a meal without giving you the drink of alcohol before that. People that drink alcohol, most people, not everybody, of course, there's always that bell curve, right? Most people will consume more food after they've had a drink. So if you're going into a big meal, have a cocktail beforehand. You'll be able to eat more of that good food. But this may actually tie back to this whole notion of transient nutritional resources in our early primate ancestors. When they were able to find a source of food, tracking it down by the plume of ethanol that it was emitting, they needed to eat. And so tying these two things together, basically identification and stimulation of feeding, kind of makes sense. There's another thing that we notice about evolution and fitness and the ability to sort of live better under specific conditions, that they hang on. When you have an advantage and you keep that advantage around, you hold on to it. When you look at epidemiological studies of humans and you look at more cardiovascular risk and mortality, low levels of ethanol consumption relative to either abstinence, not having any alcohol at all, or overconsumption, drinking way too much, you're better off drinking a little bit. It's why doctors tell you to have a glass of wine or two a day. That's basically these curves here for women and for men. When you start drinking, when you go from abstinence here, which is the baseline of the relative risk of mortality, you start drinking a little bit, you start to get an advantage in living longer. So if you think about these primate frugivores, they're going out and they're eating fruit, and basically all of that fruit is ripe, or they're trying to eat the ripe fruit. The ripe fruit has ethanol in it. So their base diet contained small amounts of ethanol. And our physiological systems may be better adapted for having small amounts of ethanol in our diet, where we're sort of used to having a little bit of ethanol in us, and that makes us function better. When people talk about the hygiene hypothesis and allergies and how we're not exposed to enough dirt anymore, it's the same sort of thing. Our bodies evolved with all sorts of parasites in us. And a lot of those parasites stay in us by turning down our immune system. They sort of like interact with our immune system and use these chemical signals to hide from our immune system. That's how they stay in us and keep our immune system at bay. And so these dialing down chemicals from these parasites actually probably are the baseline. And without all those parasites in our water and our food, because we have all this modern technology, our immune systems are kind of jacked up and we have a higher incidence of allergy. Same sort of thing, except ethanol, right? So what we're looking at here in this ethanol early hypothesis is that ethanol is a cue that fruit is ripened and is in its most nutrient-rich state. Yeast want the same sugars out of it that we want. Our early, and we're talking 80 million years ago, this is a big departure from that 10,000 year old we started agriculture and had extra food so we made beer hypothesis. This is going way back. What was one of the trivia slides earlier that said like dinosaurs went extinct 65 million years ago? We're in that range. That these arboreal primate ancestors may have used ethanol plumes to find ripe fruit. And so ethanol as a feeding stimulant and low levels of ethanol improving health are evolutionary hangovers from our primate ancestors and millions of years of evolution around alcohol. That's a lot of time that we've spent interacting with yeast and an evolutionary history that's pretty impressive. So this goes back to my original question. Where did the genes for ethanol metabolism come from? Genes don't just randomly appear. I mean, okay, we can get into bacterial genetics and how they swap genes and like stuff like that. But when we're talking about primates, we're not, just passing plasmids back and forth. We had to have had some sort of similar function. So remember I was talking about antifeedants. A very potent antifeedant is this geraniol. Sounds like geranium. And actually, for those of you that have a good hoppy beer in your hand, take a whiff. Geraniol is actually a compound, a positive aroma compound in hops. When we dry hop beers, one of the things we're looking for is for this guy to be there because it gives us that floral aroma to the beer. And so it's a positive thing these days in our beer, but when you're looking at a plant-based diet, this is one of the ways that plants try and keep organisms from eating the plant. This is one of their chemical weapons. And so there's enzymes that our primate ancestors had because they had a plant-based diet to break this guy down. Okay, so when we look at chemical structures, this little doggy, ethanol, looks a lot like this end of the geranyl molecule. And when we're talking about breaking down ethanol, we're going to oxidize this end. And when we're talking about breaking down geranyl to make it not an antifeedant, we're talking about oxidizing this end. And so you can imagine if you have an enzyme with an active site that fits this guy and oxidizes it, that fits in there pretty well too. This is a phylogenetic tree of a bunch of primates. You can see the primates are over here. Basically, the distance between any 2 points is a measurement of the distance these two organisms are from each other based on their DNA, how evolutionarily distant they are. Two things that are very close are very closely related. Things that are on opposite ends of the tree with a really big distance between them means they're very evolutionarily distant. When you have a point here, a node, where it splits off, that's basically a sort of like a speciation event where you have organisms, two organisms coming from a common ancestor. This point here is the common ancestor. And then you have the one organism splitting off and the other organism splitting off. So this is a really cool paper. I've talked a little bit about it, that hominids adapted to metabolize ethanol long before human-directed fermentation. But we're down here with this GCH group, I think is what they call it, the gorilla, chimpanzee, human. And they're looking at a single gene here, this ADH4. ADH4 is alcohol dehydrogenase 4. It's one of those enzymes that oxidizes that ethanol molecule and starts the process of breaking it down. This is the one that's primarily gastric. It's in our mouth and esophagus as well, but it's the first shot we get at breaking down ethanol. Some of the differences between the ability of women and men to consume alcohol and the effects it has on them are tied to various amounts of this enzyme. So it has a really big impact on our ability to consume alcohol. But this is a single change in the protein. You're looking at one amino acid change, alanine to valine, is that right? At this specific locus, at amino acid 294, that leads to this group down here being able to break down ethanol about 40 times better than they can break down, than this other enzyme form, this ADH4, can break down geranyl. And so basically we had the enzymes for breaking down geranyl, but at some point it became advantageous for these guys to be able to break down ethanol really well instead. And when you look at these organisms and you look at what their fallback food is, I mean, a lot of these guys are eating ripe fruit. And they have the other enzymes that help them break down ethanol in their liver and stuff like that, so they can consume the small amounts. But having an enzyme be able to increase its ability to metabolize ethanol 40-fold means that you're starting to hit the alcohol pretty hard. What are these guys doing that these guys aren't? Well, their fallback food is the fruit that's fallen to the forest floor. These guys are chewing on sticks and leaves and stuff. These guys are going after the fruit on the forest floor. Well, the fruit on the forest floor is overripe. And what do you think that means for the amount of time that the yeasts have had to get at the sugars in the ripe fruit? It's gone up, right? So the alcohol level in the fruit on the forest floor is increased from, maybe less than 1% that they're getting in the ripe fruit in the trees to a couple percent. So when you're interacting with a compound that affects health, I mean, alcohol is effectively a toxin. If we don't metabolize it, it's really not good for us. So if you have the enzymes that can break down that toxin and you're not getting wasted eating the fruit on the forest floor, and you can still move around and not get eaten, there's a definite advantage to that. And this single amino acid change in this one enzyme may be why we walk upright. This is basically what we're talking about here. We have ethanol and we have the geranyl. This is that HCG, the human chimpanzee gorilla ancestor, that node 54, this node, or this sort of spot of divergence. And when these guys did something really cool. They resurrected enzymes. So they took these sequences from these various other organisms that trace back and have this greater evolutionary distance from us, and they took the sequences of the DNA, made the enzymes, made purified enzymes, and then tested how well they broke down ethanol and geranyl. So they're measuring the ADH4 activity towards ethanol and the ADH4 activity towards geranyl. When you look at this one node, it does really, really well with ethanol. This is the version we have. This is why we're doing this right now. Okay, but those other ancestors that have different fallback foods, they're more concerned about breaking down this anti-feedant in their fallback foods. It works a little bit on geranyl, but it's not very good. But if you look at this, like the ethanol that this other enzyme form, it doesn't really break it down very well at all. So this is going back to another one of an earlier paper, the Dudley paper, the ethanol fruit ripening and the historical origins of human alcoholism in primate foodivore. Science is a great language. I actually like reading scientific papers. I'm weird. But when you look at what happens when you go from the transition from ripe to overripe, and you have the sugar content here, you can see that the overripe fruit is getting into ethanol percentages about like what I'm drinking out of this glass, okay? That's pretty stiff stuff to be consuming as food. Being able to break that down and still get the nutrition out of it, especially because as you look at this, the sugar's starting to go down as the ethanol goes up, right? The yeast are breaking down the sugars in the fruit, they're taking advantage of it, they're making the ethanol saying mine and keeping all the other microorganisms from growing there. So ethanol goes up, sugar goes down. You probably have to eat more of this to get the same, well, actually, alcohol is calories too, right? And that's one of the things that we realize, beer gut, right? Ethanol has calories too. But you have this group of organisms here with a common ancestor that have this great improvement in their ability to break down ethanol. You'll notice there's also this red up here with the II. Completely separate mutational event that led to the same change in the same amino acid that lets them drink booze too. So if you're ever out partying and you run into an II, you can be sure that they can keep up. So one of the things that is really important here that ties into this why we walk upright part of this story is this middle Miocene climactic transition happened right about the same time this mutation happened. Okay, one thing about genetic clocks, you can't exactly tie evolutionary change to the passage of time. Because as we saw in those early slides about antibiotic resistance, it's about the selection, not the mutation. Mutation happens at a pretty consistent rate, but evolution is dependent upon selection events. So when you have a big climactic transition and the environment's changing, that's a selective pressure that'll drive evolution, and you'll see evolution happen faster. It sort of happens in fits and starts. And so right around the time you have this MMCT, as they call it, you have this amino acid change that lets these early guys, our early primate ancestors, eat the fruit on the forest floor. One of the things that was happening is that in Africa, where these guys were, this climactic transition was changing the forests into like big broad-ranging forests into clumps of forest with grasslands in between. So really big change. Your food sources are all changing. So these guys, fallback food, ended up being different than these guys. These guys had a niche. They were able to eat the fruit on the forest floor that nobody else could eat because it had too much booze in it. And so that gave these guys an advantage and kept this genetic change around through selective pressure. So now we're going to transition from some of the genetics into some of the cultural changes that happened with humans. Like if you think about us still having genes today for breaking down ethanol, there's got to be sort of this uninterrupted presence of ethanol from our early primate ancestors to us today. We start to move into these cultural changes in how humans live. And one of the biggest changes was the Neolithic Revolution, where you have, we were transitioning from a hunter-gatherer lifestyle into a more sedentary lifestyle, and we start agriculture. We start taking grains and purposefully sowing seed and growing more of that grain from that seed. And this is, again, ties back into that Paleolithic diet. So that paper that I was talking about earlier that I don't like, that's the paleo thing, that's actually where the paleo diet comes from. That's what they're arguing for in that. The ethanol early version already has us consuming alcohol, right? And we know it's been a part of our diet for tens of millions of years at this point. If we're only talking about 15,000 years ago, humans and their primate ancestors have been consuming alcohol for tens of millions of years at levels not dissimilar to what we're consuming today, by the way. So when you have these groups of people settling down, why are they settling down? Why are people starting to live in one place? One of the great things about ethanol is that it's, with the enzymes to break it down, is that while it is sort of a toxin, we can break it down fast enough that it doesn't do super bad things to us. But we also are still not breaking it down fast enough that it doesn't get to our brain and do some interesting stuff to our psychology and how we perceive the world around us. And there's this euphoric sensation that you get when you drink, right? There's some people that suggest that when you start to have these psychological changes, we see the drug use in humans and other animals, ethanol consumption being one of them, that you start to tie that to ritual, and you start to have a culture around this change in your psychological state, and that can lead to things like religion. And when you start to have religion and culture and you start to then, worship stuff, you start to build things to worship that stuff at, and shrines and temples, that sort of thing. Well, okay, if you guys need some help moving, no, seriously, follow me on this one. When you guys need help moving, what do you put out on Facebook? I'll provide the pizza and beer, right? That's usually how you get people to show up. Well, when they were building these temples and stuff, you're getting people to come together to work on something. What did they do? They had parties. Alcohol was actually probably an important part of the ritual. And so basically you have people starting to come together around this ritual, which probably involved alcohol, not dissimilar from helping your friends move. But the argument then sort of transitions to, well, what were we, when we started agriculture, so we start living in one place, we start doing all this religion stuff, we start doing agriculture, why were we growing grain? There's a couple things you can do with grain. You can make bread, or you can make beer. And there's actually a really sound argument for the Neolithic Revolution not being about bread and food, but actually being about beer as food. When we look at some of the earliest drawings and writings, we see images like this. Amanda showed that early tablet. It had a vessel similar to this, straws coming out of it, people drinking from it. This is beer, a very early form of beer. It's not something we would necessarily recognize as beer. It's not processed to the level that we process beer. They didn't have like Mura mash filters like we have at Full Sail. They were basically taking grain and allowing it to sprout, drying it, and then mixing it into a gruel and then fermenting it, or allowing it to ferment. They didn't actually understand the microbiology of fermentation back then. But suppose that the consumption of a food produced an altered state of awareness, and this is getting back to that sort of psychological impact of drinking. And it's noticeable, but that you're not having a serious toxic side effect. Now suppose that this food also had a second imperceptible effect. This is one of the keys here. You have an improvement in nutritional value over the unprocessed cereal grains. Fermentation actually makes those grains more nutritious. And this is what happen when you start turning barley and wheat into beer. It becomes more nutritious than the grain itself originally was, okay? And so when we talk about the improvements in nutrition, what are we talking about? We're talking about the production of beer vitamins, vitamin B, one of which is folic acid. The yeast are growing and dividing and making protein. Some of that protein contains lysine, which for us is an essential amino acid. We can't make lysine, so we have to take it in from our diet. This yeast-laden beer is actually very rich in lysine, so we're getting a necessary vitamin, an essential amino acid, but then you also have these compounds in the grains called phytates, and they tie up minerals and prevent us from taking them into our bodies. We'll ingest these phytates, and they'll sort of bind up minerals like calcium. It's kind of important. and keep us from being able to uptake that calcium because it's bound up in this molecule that passes through. And so if you're reducing these phytates through the process of fermentation, you're not only improving nutrition, you're also making these minerals more accessible. That's a lot better than bread, plus you get a buzz. So what does it take to make beer? How do we discover the process of making beer? There's some basic steps that really aren't that far of a reach to get from wild cereals, wild grains that we're interacting with out, as we're hunting and gathering, to making beer. So you have the presence of the wild cereals, and we're using them. You're making some sort of gruel or porridge, so you're basically putting the grains into liquid and sprouting and then drying them. This is what we refer to as malting, where you're sprouting the barley and making malted barley. This releases some enzymes that start to break down the complex sugars in the seed, and because the plant is basically getting ready to use those sugars as food to grow a new plant, but we stop it from growing the new plant by drying it and fix those enzymes and those sugars and basically use those enzymes to turn that seed into a whole lot of sugar. The other thing is, so the seed is sweeter and the husk is more easily removed, and the husk is not really of any nutritional value. So somebody gets lazy, and their gruel sits around for a little while. If you saw my kitchen, I could totally have been the Neolithic guy that did this. But it doesn't spoil, okay? Yeast is sort of omnipresent in the environment. If it's on fruit and randomly moves from fruit to fruit, and it's carried by insects, and it just is in the air, quite literally, it makes its way into this gruel. And so this gruel, it doesn't spoil, but it makes beer. And so that beer is both psychoactive, it changes your mood, and it's also more nutritious than that gruel originally was. You have the vitamins, you have the essential amino acids, you're reducing the phytates and you're getting more nutrition out of it. And then because probably of the psychoactive impacts, it's making its way into rituals. So we've talked about our early primate ancestors, the transition to walking upright, but we're not quite to modern humans. Okay, so we have our friend Saccharomyces cerevisiae. We know that it's very important because some of the earliest writings are about beer. Amanda showed that tablet earlier that had these sort of drawings on it, the vessel with the marks in it, those marks indicate that vessel contains beer. We see other pictographic evidence of people drinking out of straws and making beer. Here's another good example here, the vessel with the straws. And so we have yeast, and at this point, we're creating whole societies around the consumption of alcohol. Like society itself was a construct of the consumption of alcohol. And the way I look at it, yeast basically has domesticated us. So think about, what do we use like chickens and cattle, like domesticated animals for? We use them for food, right? Well, yeast is basically using us for food. We're giving it this malted barley and wheat, giving it all the sugars it wants, and we're feeding the yeast. We're basically its cattle. It sounds a little weird, but when you think about it, the yeast had control. They were basically using us for food. And that's great. I mean, we were getting something out of it too. This is part of the whole coevolution story, is we have this intimate relationship with these guys over tens of millions of years. I think yeast won out early on, but we'll get to some changes in that in a little bit. So this ethanolic fermentation, remember when I was talking about how the production of ethanol helps the yeast keep other microorganisms from growing? The ethanol is a potent biocide. It's one of these chemical and biological weapons. We talked about some of the other antibiotics and stuff, but ethanol works really well. So the other thing that you have when you're making beer is you have to heat those the grains to get the activity of the beta and alpha amylase, the enzymes that you get from the sprouting process, to break down the complex sugars in the seed to make the simple sugars that the yeast can use. And so in addition to having a potent biological weapon as ethanol in the production of beer, you also are heating the water up. Heating water is really important to kill microbes, right? You see boil orders for, even today, for when there's like a pipe break or something like that, or somebody pees into Mount Tabor Reservoir, and they tell you to boil the water. Well, basically, in the process of making beer, you're heating the water to a point where you're killing the microbes in it. So not only are you killing the microbes in the process of processing it, and you're making the ethanol, that's a 1-2 punch for keeping stuff from growing in it, and that makes it safe to drink. Here's a great little picture from Carlsberg, India, one of their breweries in India, of their wort kettles, and of them boiling their wort, basically hitting this brewer's window, or I guess this is already after they've mashed in, and anyways. But they're boiling it again, even after they've made the sugars. So it gets even more boiling, which kills more microbes. So when you think about modern humans, and you start having this Neolithic revolution where you start having people settling down and living next to each other and not living these nomadic hunter-gatherer lifestyles, one of the side effects of that is that you're stationary, and you're stationary around a lot of other people, and people poop. It's one of the basics. And so when you think about it, you're basically always downstream of somebody. That river, that little creek, something upstream has pooped in it. And fecal-oral transmission is a really easy way to get you sick. And when you don't have antibiotics, unless you have the tetracycline beer, those guys were probably doing a really good job, you're going to get sick and die from microbial infections before the advent of antibiotics. And so if you're not drinking water, what are you drinking? You're drinking beer. And beer is safer than water. Beer has alcohol in it, and if it's one of your main sources of water and nutrition, then it actually becomes really important. So we have people living in more and more concentrated groups. People are starting to live in cities. One of the big cities in the world is London. And you have cholera outbreaks in London. This is a great example. This is another slide that I like to use in my microbiology class to talk about epidemiology. So epidemiology is the study of populations and disease. And this guy, John Snow, is credited with the first epidemiological study. And he basically went around to all the houses during this cholera outbreak in this area and marked each cholera death as like a little black bar on this map. And the black bars are about this wide. See that one right there? That's like one death. This is a lot of dead people. And he co-located these deaths with this Broad Street pump. And this was a pump for water that the people in the area would go to get their drinking water. And when he did this interesting study, he looked at the other pumps. They're marked here. And some of these people that were distant and still died, or were closer to other pumps, they actually liked the water from the Broad Street pump better, so they went out of their way to go there to get water. Or they'd been passing through the area and gotten water there, and that's why they ended up dying of cholera. There's one thing to note here. Okay, so you're looking at all of these buildings right around this pump, and they all have a lot of dead people in them, except for one. See this guy right here? Probably can't read what that says. There's an exception to all of these deaths in this one place. What do you think that place is? It's a brewery. So much the way that I go to the pub at Full Sail after work and get a couple free beers, my shift beers, it's a thing, it's awesome. It's one of the best perks of the job. These guys got beer as part of their work, as their daily ration. They weren't drinking the water from this pump like a block over. They were drinking the beer instead. They all lived. Everybody around them was dying, and they lived because beer was safer than water. And it's evidenced in the first epidemiological study ever. I never knew this when I was teaching in my microbiology class. I always just talked about, the co-location with the pump and everything. I never got into the story deep enough to realize that's a brewery there with no dead people in it. I think my students would have really liked that one. So that brings us basically up to modern humanity. We've talked about our primate ancestors in the trees using ethanol to find fruit. We've talked about us transitioning to walking because of a single mutation in the alcohol dehydrogenase 4 enzyme that allowed us to eat the overripe fruit on the forest floor. And we've talked about settling down into agriculture, and now we get our beer. Okay, good pacing. So, what about the yeast? We've talked a lot about the genetic changes in human. We've talked about the ability to break down ethanol probably coming out of enzyme changes in things that broke down antifeedants to breaking down ethanol. We've talked about holding on to these genes that break down ethanol because beer was really important to us early on in the Neolithic Revolution. And we held on to those genes because beer is safer to drink than water through much of history until we get modern drinking water treatment, which really didn't happen until the last 150 years. And so, what about the yeast? When the yeast have changed too, and the yeast were changing before we even knew there was such a thing as yeast. They're sort of our companions in this 80 million year journey. And we didn't know they existed until this fabric merchant, Leo Windhoek, designed an early microscope and looked at beer. So this is his drawings of yeast. You can see it's about beer, globs of stuff in beer. These globs are our friends up here, Saccharomyces cerevisiae. So he was actually able to visualize these guys using a microscope that fits in his hand. Think about that for a second. Think about what our microscopes look like and think about what he was able to do with this thing that fit in his pocket. Oh, fabric merchant, right? So he was looking at thread counts basically with this and then started looking at other stuff and basically was, along with Hooke, is sort of credited with the discovery of microbiology. He looked at everything. Read up on this guy sometime, he's a really interesting character. But one of the things that he looked at, in addition to like poop and stuff, was beer. And he used his microscope to visualize yeast. And so there was this huge argument about what causes fermentation, how fermentation happens. And a lot of the chemists were like, it's a chemical process. It happens, there's this transition, the exposure to oxygen, all this stuff leads to the production of alcohol and CO2, and it's a chemical process. Then there was this guy that came along, Pasteur. So he didn't discover that yeast were microbes, but he actually proved to most people at the time that yeast are the active agent in fermentation. And so I'll let him do the speaking. The chemical changes of fermentation are associated with a vital activity, beginning and ending with the latter. I believe that alcoholic fermentation never occurs without either the simultaneous organization, development, and multiplication of cells, or the continued life of cells already formed. He's basically saying, these guys here are what makes the ethanol happen. That it's a biological process, not a chemical process. If you cheat and take the enzymes out of these guys, you can make it happen as a chemical process, but don't tell those chemists. It's a biological process. And that's pretty cool. Now we know what yeast are. But we've been making beer for 10,000 years probably, 15,000 years. We don't really even know. We've been making beer for a really long time. One of the funny things about grains when you malt them, remember I said that husk comes off easily? That husk protects the grains in archaeological sites. And so if you're malting the grain, you might never see the grain in archaeological sites. If you carbonize them, if you burn seeds, you can also find them in archaeological sites, but while there's heat involved in the process of making beer, you're not directly heating the grains. So you're not going to find carbonized grains. So we don't really know how long we've been making beer because we don't have the archaeological evidence to demonstrate what these sprouted grains, like, you're not going to see them. And so we've been making beer for 10, 15,000 years at this point, and all of a sudden we figure out, oh, hey, we've got some friends, and these friends have been helping us do this the whole time. And, people had an idea that this was going on. It's not like Pasteur figured out that, oh, hey, there's yeast in this. People knew that there was a process going on here. And when we make beer today, I was just doing this earlier today, we take yeast out of one fermentation, count the cells, check the viability, and then throw that into the sugary wort to make more beer. People have been doing that for probably almost as long as there has been beer, where basically they take a little bit of beer from one batch, put that into the sugar that they're making the next batch from, and it comes out like the last batch. That's probably how those guys kept that tetracycline going in their beers, because they were using some of the microbiology from the previous batches to make the next batch. And so if you keep doing that, you're basically keeping that one group of yeast going. And when you're putting yeast into the environment of brewing, you're applying selective pressure. So we are actually causing yeast to evolve by making them brew beer. So we've talked about humans, we've talked about yeast, we've talked about the ties that we have to yeast going all the way back to where the origins of controlled fermentation happened, where humans started to settle down, the differences between wine and some of the history of the beer strains. And so yeast are basically our friends in evolution from 80 million years ago and through to today. And we see us mirrored in them in some really interesting ways in their genetics. And so we truly have co-evolved as these two species, yeast and humans, and it's all centered around our desire to drink alcohol. So with that, I'll close up, and thank you very much.
Thank you for listening. This podcast and Science on Tap events are created by Via Productions, and we are based in Portland, Oregon in the U.S. If you'd like to find out more about how to attend one of our events, check out scienceontaporwa.org, and that last part stands for Oregon and Washington. You can also find us on Facebook, Twitter, and Instagram at scienceontaporwa. As always, I'd like to say thank you to my volunteers, affectionately known as the Minions. By the way, they were called that way before those little yellow Twinkies became popular. Anyway, they've been helping me run events for years, and I could not do this without them. The Minions are Scott Fry, Chris Gowan, Sam Lauk, Rita Nygren, and Stephen Perry, as well as many other people who have helped out over the years. I'd also like to say a special thank you to Amber Peoples for stepping in and running things for the past few months. You all are amazing. Also, a final thank you to Jonathan Coulton for letting us use his song Mandelbrot Set as our theme music.
Just